we will begin our section on cellular respiration. In this first part, we'll do an overview of cellular respiration before we dive into some of the details. Cells extract energy from food to generate ATP that is then going to be used to run different reactions in cells, such as muscle contraction. Typically, cells will prefer to use glucose catabolism in a process known as cellular respiration, where the energy that is contained within the sugar molecules is broken down and released for the formation of ATP. Here we see glucose in the presence of oxygen being broken down into carbon dioxide, water, and energy in the form of ATP. Now we also know that in metabolic pathways, this link between the transfer of energy from one molecule to another often follows the transfer of electrons. Electrons have high energy levels, and when we pass electrons, we pass that energy. We often pass this energy in an incremental fashion. If we passed all of it at one time, we would tend to lose a lot of the energy as heat. But if we can pass it in small packets, we can extract more of that energy in a usable form. So what we'll often see is these electrons being taken off of these molecules like sugar and gradually reduced in their energy level in such a way that we can extract that energy off rather than just dropping it all the way down and losing a lot of that energy as heat. So we will take glucose. In the end, we'll have the formation of water. We'll end up with carbon dioxide, and all of that energy that we've extracted can then be used to make ATP. Let's take a look at the concept of redox reactions, because that's what these are. These are reduction-oxidation reactions, also known as redox reactions. Redox reactions are where electrons are transferred from one molecule to another. You've probably learned about this in inorganic chemistry. In a redox reaction, we have one molecule that will be oxidized and another will be reduced. And then we also have a reducing agent and an oxidizing agent. Reducing agents are molecules that can donate electrons in this redox reaction, so they can give up electrons. When they give up those electrons, they become oxidized. The oxidizing agents are the ones that can accept those electrons. So one gives them up, the other one accepts them. So the reducing agent gives it up, the oxidizing agent takes it. The reducing agent has become oxidized, the oxidizing agent has become reduced. And in biological systems, this transfer of electrons is typically in the form of hydrogen. So here we have glucose. It has a lot of hydrogens in it. When we go through cellular respiration, eventually we're going to take that glucose all the way down to carbon dioxide and water. When I take these hydrogens off of this glucose, I'm oxidizing it. Molecules that lose electrons are oxidized. I oxidize glucose to carbon dioxide where the hydrogens have been removed. This oxygen is going to pick up those electrons and form water. So oxygen is reduced to form water. So what was the oxidizing agent here? It was oxygen, because oxygen picked up the electrons. The oxidizing agent has become reduced. What was the reducing agent? The reducing agent was the one that donated the electrons. So it was the glucose. It was the reducing agent which became oxidized. Now this doesn't happen all at once. There's many steps that we're going to see in this chapter to get us to this overall general reaction. And glucose is not going to just directly hand all of these hydrogens over to oxygen. It's going to do it using what we call electron carriers. So what are electron carriers? This is an example of an electron carrier, nicotinamide adenine dinucleotide. It is a dinucleotide, so we have our 5-carbon sugar, a nitrogenous base. There's 2 of them together, so this is the dinucleotide. And it can exist as either a reduced form or an oxidized form. So over here where the hydrogen's been reduced, it's in its oxidized form. Over here where it has an extra hydrogen right there, it's in its reduced form. And it can go back and forth between these two states. So the NADH over here is going to carry more electrons and, of course, another hydrogen than the NAD. So that's the reduced form of it. And it's called an electron carrier because it can accept electrons from other molecules. And then, of course, NADH can donate electrons. So it can both receive them if it's NAD, and it can donate them if it's NADH. These molecules are very important in reactions like cellular respiration, photosynthesis, because these carriers here will shuttle these electrons over to an electron transport chain. And then it's in the electron transport chain where we'll actually make ATP. So we're holding on to all these electrons that we're later going to use to make ATP. There's other electron carriers. We have FAD and FADH2, not quite as high an energy molecule as NAD, so the energy that we get from this carrier isn't quite as high. And then we also have NADP and NADPH. This is found in photosynthesis. Let's talk about ATP for a moment. ATP is, remember, that energy currency that we find in living systems. This is what's going to supply energy or allow us to store energy in biological systems. And we use it in coupling reactions. So what we see down here is we see a molecule that we are going to energize. And we're going to energize this molecule by taking ATP, taking that phosphate group off, handing it to this molecule, and that's what we see over here, is that phosphorylated molecule. This molecule now has gained some energy. Now, of course, ATP has lost some energy when it did that, so this went down in energy level. This was an exergonic reaction. Here, the ATP to ADP was an exergonic reaction. And it drove this other reaction here, which was an endergonic reaction. It required energy to energize this molecule up here. Where did we get the energy? We got it from the hydrolysis of ATP. So we coupled those two reactions. So when we do this, we have these two different terms that we use, dephosphorylation and phosphorylation. So in dephosphorylation, we are going to take phosphate off of a molecule. So right here, ATP was dephosphorylated because we took a phosphate off of it. Remember, ATP is adenosine triphosphate, ADP is adenosine diphosphate. So we took a phosphate off. That was a dephosphorylation reaction. The same time that we did that, we ran a phosphorylation reaction. We coupled it. What we phosphorylated was this molecule here. So we took this molecule, we added the phosphate to it over here, so that reaction was a phosphorylation reaction. So typically we're going to phosphorylate and dephosphorylate in these coupled reactions. Now, phosphorylated molecules tend to be less stable and they have a lot more energy. So whenever you're looking at these metabolic reactions, when you see a molecule gain a phosphate group, it has gone up in energy level. And when you see a molecule lose a phosphate group, it's gone down in energy level. This molecule here on the right has a much higher energy level than the molecule here on the left. Likewise, ADP has a much lower energy level than ATP did. So as we saw before in overview, when we look at cellular respiration, and this is aerobic cellular respiration we're looking at right now. So aerobic means that it is in the presence of oxygen. That's what aerobic means. So in the presence of oxygen, we're going to take glucose and we're going to break it down into carbon dioxide and water. And if we are in an aerobic pathway, we are going to completely break down the sugar. We're going to see other pathways where there's a partial breakdown. If there's a partial breakdown, what happens is we don't get all of the hydrogens off of this sugar, so we're left with some hydrocarbons left over. But here, we've extracted all of the hydrogen out of there down to CO2 and water, so that's a complete breakdown. Once again, partial breakdown, we would have some sort of a hydrocarbon left over here on our product side. Now, to be able to do this is going to be many steps, as we've indicated before. So we're going to see lots of different steps of cellular respiration. Let's just briefly take an overview of those steps. Stage 1 is glycolysis. Then we have stage 2, which really consists of two steps here. The oxidation of pyruvate and the formation of acetyl-CoA. And then we have the citric acid cycle. So those are both considered stage 2. And then stage 3, we have oxidative phosphorylation. So we're going to take a look at each one of these stages independently. Let's first take a look at this video that gives us an overview of the stages of cellular respiration.
This bird, like all living animals, has cells that carry out cellular respiration. During cellular respiration, organisms break down nutrients in order to produce energy in the form of ATP. Let's take a closer look to see how this happens. If we zoom in on one of this bird's cells, we can see the mitochondria, where the majority of the reactions involved in cellular respiration occur. Let's zoom in further on a single mitochondrium. Cellular respiration consists of 4 primary stages. These are glycolysis, the preparatory reaction, the citric acid cycle, and the electron transport chain. The first stage, glycolysis, occurs in the cytoplasm outside of the mitochondria. During glycolysis, enzymes break down glucose into 2 molecules of pyruvate. During this process, 2 ATP are produced, and NADH is released to be used in a later step of cellular respiration. Stage 2 is the preparatory reaction. During this stage, the pyruvates of glycolysis move into the mitochondria, where they are converted into acetyl-CoA. During this conversion process, more NADH is produced and carbon dioxide is released. In stage 3, the citric acid cycle, the remaining carbons from the initial glucose are oxidized, releasing carbon dioxide. NADH and FADH2 are also produced, in addition to 2 ATP molecules. Most ATP production occurs in stage 4, the electron transport chain, or ETC. In this stage, the NADH and FADH2 from the previous stages give up electrons to the chain. Energy is released and captured as the electrons move from a higher energy to a lower energy state using a series of proteins embedded in the membranes of the mitochondrion. Later, this energy will be used for the production of ATP, typically 32 to 34 per initial glucose. Oxygen is used by the ETC as a terminal electron acceptor. It then combines with hydrogen ions to produce water. If we add up all the ATP produced from just one glucose molecule, we can see that these reactions produce a total of 36 to 38 ATP.